A unique type of RNA viruses are the retroviruses. So we're going to talk about some of their unique features, and we're going to look at the life cycle of a retrovirus using HIV as an example. Now, it's important to understand that among the RNA viruses, there are three main general strategies: single-stranded RNA viruses, double-stranded RNA viruses, and then the retroviruses. These first two, the single-stranded and double-stranded RNA viruses, we talked about in an earlier video, have to bring with them an RNA replicase, right? They have to have with them an RNA-dependent RNA polymerase, because the one that's found in the human cells or any other eukaryotic cell is going to be DNA-dependent and will be useless for replicating that RNA. The retroviruses are a unique group, and they really do behave in their own way. So let's take a look at how a retrovirus works. We'll start with HIV. And we'll use HIV as our example, right? And you'll see that it's a variation on the theme of the life cycle of the viruses that we've already talked about. So entrance, this is when attachment penetration takes place. Retroviruses tend to be enveloped viruses, so we're going to see membrane fusion taking place. In the case of HIV, the target cell are what are called CD4 T cells. It's an important subset of white blood cells. And there are CD4 proteins on the surface that attachment spikes on the surface of the HIV virion are going to interact with. So we're going to see membrane fusion with the host cell membrane. Uncoating needs to take place, and that's going to take place right at the membrane. So the nucleocapsid needs to get degraded. The membrane gets degraded, the capsid gets degraded, and it turns out there's a capsid inside the capsid, and we call that a core. So for some of these retroviruses like HIV, there are additional proteins protecting the nucleic acid, the RNA. Then reverse transcription has to take place. And we're going to look at an image of this on the next slide, and it'll make a lot more sense. But the retroviruses are single-stranded RNA viruses. But what happens is, using an enzyme called reverse transcriptase, a double-stranded DNA copy of that single-stranded RNA gets made. That's the reverse of transcription, right? Transcription is a double-stranded DNA being copied into a single-stranded RNA. Reverse transcription is a single-stranded RNA being copied into a double-stranded DNA. That double-stranded DNA enters the nucleus, and an integrase enzyme is going to randomly insert it into the genome. What does that sound like? That's a lot like, at least analogous to, a lysogenic phage lysogenizing its host cell. Now, we don't use the term lysogeny to talk about retroviruses, but conceptually it's very similar. So we've got two unique enzymes so far, reverse transcriptase and integrase, and now our double-stranded DNA is randomly inserted into our poor little CD4 T cell's genome, meaning that it's there permanently. So as long as that CD4 T cell is alive, it has the potential to produce new variants. This is why it's so difficult to fight some of these retroviral diseases like HIV, because we can chase off, we can kill, we can inhibit and eliminate whole HIV virions, but if there's always a genetic source in these CD4 T cells, it's practically impossible, without killing off all their CD4 T cells, to actually get rid of ultimately the source of the virions. It becomes a persistent infection. At some point, transcription of the inserted double-stranded DNA is done and viral messenger RNA is produced. Then we get assembly of all that, the proteins and mRNA or RNA that was produced, and encapsulation. We call this maturation. There's a key protease involved, and I mention it because that protease can be inhibited by certain drugs, and so many of our anti-HIV drugs are in fact protease inhibitors. And then because it's membrane bound, it's an enveloped virus, it has to release by budding, like we talked about. So let's take a look at what that looks like. Up here in the top left, you see an HIV virion. It has spikes, and its spikes are made up of two proteins. The proteins are called GP41, or glycoprotein 41, and GP120, glycoprotein 120. So right here, you've got a combination of GP41 and glycoprotein 120. The GP120 interacts with a protein on the surface of the CD4 T cells called CD4. And so GP120 hits CD4, and that triggers an endocytosis, and then you've got this crazy double membrane that has to be broken apart. And the way that happens is with the GP41 portion of the spike actually fuses those two membranes together, does some gymnastics, and allows the nucleocapsid to be exposed and uncoated. And then the RNA gets synthesized into DNA. It gets reverse transcribed into DNA, and the DNA, double-stranded DNA copy of the genome, enters the nucleus, where the integrase enzyme integrates it randomly into the human DNA, and it becomes what's called a provirus. Just like we saw with lysogeny, we talked about the prophage being the DNA in the bacterial chromosome. Here we've got a provirus, which is the HIV DNA inside the CD4 T cell genome. It can sit there quietly for years and years and years. We call this latency if a virus is sitting quietly. Or it can start transcribing right away. And if it starts transcribing right away, the messenger RNAs are exported and translated into all the various proteins. So what proteins do we have? Well, you've got core proteins, you've got capsid proteins, and of course you have to put some GP41 and GP120 into the host cell membrane so that when budding takes place, the bit of membrane that is captured has spikes on it so that it can start the process over with a new CD4 T cell. It's a slight variation on the lytic and lysogenic cycles that we saw in bacteriophage, but you can see conceptually that it's a very similar process. So let's summarize our retroviruses here. They're enveloped single-stranded RNA viruses with a very unusual life cycle. They use reverse transcriptase and integrase to insert a double-stranded DNA copy of their genome into the host genome. And the provirus then, the DNA of the HIV that is now integrated into the CD4 T cells chromosome, becomes a long-term source of virions leading to a chronic infection. Thanks for joining me, and I will see you next time.